In this lecture, we'll focus on the metabolism of amino acids and other nitrogen compounds. In addition, we'll study the urea cycle, which is the major pathway for the elimination of excess nitrogen from the body. In humans, amino acids are derived from three sources. During passage through the digestive system, dietary proteins are subjected to enzymatic hydrolysis. This releases the 20 component amino acids and makes them available for catabolic or biosynthetic pathways. There's a constant turnover of cellular proteins. In this process, pre-existing proteins are subjected to proteolysis, and the component amino acids are scavenged for catabolic or biosynthetic pathways. About 8 to 10 of the common amino acids must be provided in the diet because humans lack the metabolic pathways for their biosynthesis. The remaining amino acids can be synthesized from simple building blocks. The digestion and absorption of dietary proteins occurs in the gastrointestinal tract. In the stomach, proteins are denatured by the acidic environment and digestion begins. This preliminary digestion is catalyzed by the proteolytic enzyme pepsin. Pepsin cleavage yields a variety of peptides, but no free amino acids are released. The majority of protein digestion occurs in the lumen of the small intestine. The intestinal lumen contains numbers of enzymes that attack proteins. These intestinal proteases are secreted by the pancreas as inactive zymogens and become activated when they reach the intestine. Proteolytic attack in the lumen reduces proteins to amino acids and smaller oligopolypeptides. Other proteolytic enzymes which are attached to the luminal face of intestinal cells reduce the smaller oligopeptides to single amino acids or to di or tripeptides. Various transport systems transfer amino acids and di and tripeptides from the lumen of the intestine to the interior of intestinal cells. Here the peptides are cleaved into individual amino acids. The amino acids are then released into the blood for delivery to the tissues. The figure illustrates the progressive digestion of protein into amino acids, first in the intestinal lumen, then on the exterior face of intestinal cells, and finally in the interior of intestinal cells. Essential amino acids must be provided in the diet because humans lack the enzyme pathways for their biosynthesis. There are 8 amino acids that are clearly essential. The amino acid histidine may also be essential in humans, but this requirement is difficult to demonstrate. 10 or 11 amino acids can be synthesized by humans and are generally considered to be non-essential. However, 2 of the non-essential amino acids, cysteine and tyrosine, can only be synthesized using essential amino acids as starting materials. In addition, some of the non-essential amino acids, for example arginine, may be required in the diet under certain conditions where the metabolic pathways cannot synthesize them fast enough. Generally, the non-essential amino acids are small and are constructed by short pathways. The essential amino acids are larger and are synthesized by more complicated pathways. These 8 amino acids are unequivocally essential. They are totally, clearly, completely essential in the human diet. This figure shows that the non-essential amino acids, coded in blue, generally have shorter biosynthetic pathways than do the essential amino acids, which are coded in red. Dietary and recycled amino acids can serve a variety of functions. They can be used directly for protein synthesis. Amino acids are not stored, so the dietary excess is degraded. The nitrogen content can be used as a source of nitrogen for synthesis of other amino acids or for other nitrogenous compounds such as nucleotides or amino sugars. Excess nitrogen is converted into urea and excreted. The carbon skeletons can be converted to acetyl-CoA, TCA cycle intermediates, or glucose and glycogen and used for energy. On the right side of the figure, we see the pathway of protein catabolism. The general pattern of protein utilization is breakdown of proteins into individual amino acids by the action of proteases, removal of the nitrogen atoms from the amino acids by transaminases, amidases, and other enzymes, conversion of the amino acid carbon skeletons into central metabolites such as acetyl-CoA, pyruvate, and TCA cycle intermediates, utilization of these products for energy production or for gluconeogenesis. There's a definite sequence of events in the catabolism of amino acids. The first step in the process is the removal of nitrogen atoms. Once the nitrogen atoms are removed, the carbon skeletons of the amino acids are subject to catabolic processes which convert them into intermediates of glycolysis or the TCA cycle. The fate of the nitrogen atoms varies depending on the organism. In many higher organisms, there's no specific mechanism for nitrogen storage, so the excess nitrogen has to be removed by some excretory mechanism. In mammals, the major excretory product of nitrogen catabolism is urea. In birds, the end product is uric acid, and in fish, it's ammonium ion. The catabolism of amino acid carbon skeletons varies in complexity from simple one-step reactions to pathways that can involve 10 or more reactions. In almost every case, the end product of these catabolic reactions are intermediates of glycolysis or the TCA cycle, specifically pyruvate, acetyl-CoA, oxaloacetate, or alpha-ketoglutarate. There are three aspects of amino acid nitrogen catabolism. One is the movement of alpha-amino nitrogen atoms from one amino acid to the other. This process is accomplished by a bevy of transaminase enzymes. Many of these transaminases involve glutamate or aspartate as one of the amino acid substrates. These two amino acids are at the core branch points of nitrogen metabolism, and they can be used to funnel nitrogen into a large variety of other compounds. A second key process in nitrogen metabolism is catalyzed by glutamate dehydrogenase. This enzyme reaction converts the organic alpha-amino nitrogen of glutamate into ammonium ion. There are only a few enzymes which can accomplish this task, and glutamate dehydrogenase is the most central. It can function in tandem with a variety of transaminase enzymes to convert the alpha-amino nitrogen atoms from any of the 20 common amino acids into ammonium ions. A third critical element in nitrogen catabolism is the excretion of excess nitrogen as urea. Ammonia is a toxic base which cannot be allowed to accumulate in the human body. The ammonia nitrogen, as well as a nitrogen atom from aspartate, is converted into urea by the urea cycle, and then the urea is excreted in the urine. Glutamate dehydrogenase is a multifunctional enzyme which helps to move ammonia nitrogen to and from a variety of organic compounds. The enzyme converts the alpha-amino nitrogen of glutamate into ammonium ion. It serves an important role in amino acid catabolism. The enzyme, working together with transaminases, can convert alpha-amino nitrogen from the 20 common amino acids into ammonium ions. It can also function in an anabolic capacity. It can convert ammonia nitrogen into alpha-amino nitrogen for the biosynthesis of the 20 amino acids. The enzyme catalyzes an oxidation-reduction reaction. The conversion of glutamate to alpha-ketoglutarate is an oxidation. The glutamate amino group is at the oxidation level of an alcohol, whereas the alpha-keto group of alpha-ketoglutarate is at the ketone level. The cofactor for this interconversion is either NAD+ or NADP+. This is one of the very few reactions that can use either cofactor interchangeably. This flexibility and cofactor specificity reflects the fact that the enzyme is functioning in a catabolic capacity when it operates from left to right, converting glutamate to alpha-ketoglutarate and ammonium ion, and is functioning in an anabolic capacity when it operates from right to left in the conversion of alpha-ketoglutarate and ammonium ion into glutamate. Having seen how glutamate dehydrogenase reaction serves as an entry and exit point between organic and inorganic nitrogen, we will now consider how nitrogen can be moved between the alpha amino groups of different amino acids. Aminotransferase enzymes employ the cofactor pyridoxal phosphate to move alpha amino groups from one amino acid to another. The transaminase process involves the formation of a covalent complex between an amino acid 1, and the pyridoxal phosphate coenzyme. That covalent complex is called a Schiff's base after its discoverer, Dr. Schiff. Certain rearrangements occur in this Schiff base intermediate. The end result is that an alpha keto acid 1, is released with a concomitant formation of pyridoxamine phosphate. The pyridoxamine phosphate is now carrying the amino group that was on the incoming amino acid. This is half of the overall transamination process. In the second half of the process, a different alpha keto acid 2, floats in and complexes with the pyridoxamine phosphate form of the coenzyme, forming a Schiff base with the amino group of the pyridoxamine phosphate. That Schiff base rearranges, and eventually an amino acid 2, is released, and the enzyme reverts to its original form. Transaminases use pyridoxal phosphate as a cofactor. The vitamin form of this coenzyme is shown on the top of the slide. It's pyridoxine, vitamin B6, a pyridine derivative which consists of a heterocyclic aromatic ring containing 5 carbons and 1 nitrogen. There are 4 substituents on the pyridine ring, a methyl group, hydroxyl group, and 2 hydroxymethyl groups. The vitamin, which must be obtained in the diet, can be converted into either of the 2 coenzyme forms, pyridoxal phosphate, which bears an aldehyde function, or pyridoxamine phosphate, which carries an amino group. Now we'll go through this process one step at a time to reinforce your confusion. In step 1, we're going to throw you the first curveball. The coenzyme is not present as pyridoxal phosphate. It's actually covalently attached to the enzyme as a Schiff's base. What was the aldehyde group of pyridoxal phosphate is linked to an epsilon amino group of an enzyme lysine residue. Now when amino acid 1 comes in, it reacts with the enzyme and displaces the coenzyme from the lysine group. The amino acid becomes covalently attached to the coenzyme in the form of an aldamine derivative. The aldamine is really a Schiff base in which the amino acid is covalently bonded to the pyridoxal phosphate. Here are a series of reactions that occur on the Schiff base intermediate. The key point is that there's a nice positively charged nitrogen at the bottom of the pyridine ring. That nitrogen atom provides an excellent reservoir in which extra electrons can find a comfortable home. What we now have is a series of electron movements. A pair of electrons move down from the alpha carbon of the amino acid and end up between the alpha carbon and the pyridoxamine nitrogen atom. A pair of electrons from the double bond beneath that nitrogen atom then moves down between the pyridoxamine carbon and the aromatic ring. Then a series of bond switches in the ring adds 2 electrons to the ring nitrogen, neutralizing its positive charge. That gives the quinoid intermediate. One important result of this rearrangement is that there is now a double bond between the alpha carbon and the alpha amino group of the amino acid substrate. In the next step, the ring nitrogen loses that pair of electrons, and there's a rearrangement of the quinoid electrons so that the double bond between the pyridine ring and the adjacent carbon atom becomes a single bond. In the process, a hydrogen atom is added to that adjacent carbon atom. The product is called a ketamine. Next, the elements of water are added to the double bond between the alpha carbon and alpha amino group of the amino acid. That addition results in the release of an alpha keto acid 1, and the formation of the pyridoxamine form of the coenzyme. This completes half of the transamination process, the conversion of an alpha amino acid 1 to alpha keto acid 1. Now if alpha keto acid 1 floats off and a different alpha keto acid 2 diffuses in, then this whole process can run in reverse. The net result of that would be that an alpha keto acid 2 is converted to alpha amino acid 2. As we noted before, this whole transamination process provides a mechanism by which the alpha amino group of one amino acid is transferred to an alpha keto acid to form a different amino acid. The original amino acid is converted into an alpha keto acid, and the original alpha keto acid is converted into an amino acid. Pass the gravy and the nitrogen. We'll now begin a short survey of other important metabolic reactions which involve amino acids. The transamination reaction shown here involves the amino acids alanine and glutamate. It's really a bridge reaction between anabolic and catabolic processes. You can think of it as a way of removing the alpha amino nitrogen from alanine to form the glycolysis and gluconeogenesis intermediate pyruvate, or you can visualize it as a way of synthesizing alanine from a glycolysis intermediate. Both of these functions will occur depending on the metabolic status of the cell. Another facet of this reaction scheme is that it highlights the important role of glutamate as a source of alpha amino nitrogen. Glutamate can be formed from alpha-ketoglutarate and ammonia in the glutamate dehydrogenase reaction. It can then go on to donate that alpha amino nitrogen to form any of the other 19 alpha amino acids in transamination reactions. Here's another short series of reactions by which the carbon skeletons of the common amino acids asparagine and aspartate can enter the TCA cycle. Asparagine can be converted to aspartate in a reaction catalyzed by asparaginase. The amide group of asparagine is removed as ammonium ion. The ammonium ion from that reaction can enter the urea cycle or be converted into organic nitrogen by glutamate dehydrogenase. The aspartate can also be converted back to asparagine by an ATP-dependent synthase reaction, not shown here. So this pathway is reversible. The aspartate can also go on to enter the TCA cycle. The end result is that asparagine is connected indirectly through aspartate to the TCA cycle. Aspartate is connected directly to the TCA cycle by means of a transaminase reaction. The transaminase converts aspartate to oxaloacetate. This is another example of an anabolic/catabolic link between an amino acid and an intermediate of the TCA cycle. The other pair of compounds involved in the transamination reaction is glutamate and alpha-ketoglutarate. Here's a parallel example in which the carbon skeleton of an amide amino acid can enter the TCA cycle. The amide group of glutamine is removed as ammonium ion by the action of glutaminase. Again, the ammonium ion can enter the urea cycle or be converted into organic nitrogen. This step can also be reversed by an ATP-dependent enzyme, glutamine synthase, not shown. The other product, glutamate, can have many fates. It can be converted to alpha-ketoglutarate by glutamate dehydrogenase, as shown here, or it can yield the same product in a transamination reaction. In addition, it can function as a precursor of a number of other biological intermediates. Biological compounds can be classified as glucogenic or ketogenic. A glucogenic intermediate can be metabolized to yield glucose as a metabolic end product. Glucogenic compounds include any intermediate in glycolysis or the TCA cycle, lactate, glycerol, and some amino acids. A ketogenic intermediate cannot be metabolized to yield glucose as a metabolic end product. The ketogenic intermediates are unable to yield glucose because they're metabolized to yield acetyl-CoA, and humans and most other animals lack the ability to convert acetyl-CoA into glucose. Ketogenic compounds include acetate, acetyl-CoA, ketone bodies, and some amino acids. Here's an example of a complicated catabolic pathway which you do not have to memorize. We use it simply to illustrate the fact that the carbon skeletons of amino acids can be either ketogenic, yielding acetyl-CoA or some related intermediate which cannot be converted to glucose, or glucogenic, yielding pyruvate, oxaloacetate, alpha-ketoglutarate, or some other intermediate which can be converted to glucose. The amino acid phenylalanine can be converted to tyrosine in a one-step reaction. The tyrosine can be catabolized in a very complicated pathway, of which only some of the steps are shown. The point is that when the dust settles, the products of the pathway are fumarate and acetoacetate. Fumarate is a TCA cycle intermediate which can yield oxaloacetate, which can yield pyruvate, which can yield glucose by gluconeogenesis. Thus, fumarate is glucogenic. The other product is acetoacetate, which can yield acetyl-CoA, but cannot yield any intermediate of the TCA cycle or glycolysis. Because acetyl-CoA cannot yield glucose, the acetoacetate is ketogenic. The take-home message is that phenylalanine and tyrosine are both glucogenic and ketogenic. This figure shows the catabolic fates of all of the 20 common amino acids. The amino acids are color-coded in purple and orange boxes. The carbon skeletons from the amino acids shown in the orange boxes yield either acetyl-CoA or acetoacetyl-CoA as the final catabolic products. These compounds cannot produce glucose, and the amino acids are listed as ketogenic. The carbon skeletons from the amino acids shown in the purple boxes yield either TCA cycle intermediates or pyruvate as final catabolic products. All of these compounds can produce glucose or glycogen. The amino acids in purple boxes are thus listed as glucogenic. Protein degradation within cells is initiated by attachment of a small protein called ubiquitin. Damaged proteins and proteins present in excess amounts are marked for destruction by the covalent attachment of multiple copies of ubiquitin. Ubiquitin attachment requires the participation of 3 enzymes. The polyubiquitinated protein is then degraded by a large ATP-dependent protein complex called the proteasome. There are 2 important features of the ubiquitin protein. It has an extended linear string of amino acids at the carboxyl terminal end. It's the terminal carboxyl group that becomes covalently linked to other proteins. Ubiquitin also has an exposed lysine residue at position 48. This lysine 48 can be linked to other ubiquitin molecules to form an extended polymer. The carboxyl terminal group of ubiquitin forms a covalent bond called an isopeptide bond to lysine residues on proteins. It takes 3 enzymes to attach ubiquitin to a target protein. The first enzyme, E1, catalyzes a reaction between ubiquitin and ATP to form an AMP ubiquitin intermediate. The E1 enzyme then transfers the ubiquitin from AMP to one of its own cysteine residues. The carboxyl terminal end of ubiquitin is now attached to E1 by a thioester linkage. The E1 enzyme then transfers the ubiquitin from itself to enzyme E2. The linkage to enzyme E2 is also a thioester. The E2 ubiquitin complex then attaches to enzyme E3. Enzyme E3 also binds the target protein. The E3 enzyme catalyzes the transfer of ubiquitin from its own cysteine residue to a lysine residue on the target protein. The final product has an isopeptide bond between the terminal carboxyl group of ubiquitin and a lysine residue of the target protein. Target proteins ultimately become attached to 4 ubiquitin molecules, which are linked to each other by isopeptide bonds. It's this polyubiquitin complex that acts as a signal to the cell's protein degrading mechanism. Studies in yeast have revealed that certain N-terminal amino acids influence the rate of protein turnover. For example, N-terminal glutamate or glutamine residues seem to increase the rate of protein turnover. In contrast, N-terminal alanine or cysteine seem to be associated with a slower rate of protein turnover. The 26S proteasome catalyzes the degradation of polyubiquitinated target proteins. A 19S cap is attached to each end of the proteolytic complex. In between the 2 caps is the 20S catalytic unit in which the target protein is degraded. The 19S cap units react with polyubiquitinated target proteins to allow the target protein access to the hollow center core of the catalytic complex. The interior of the hollow 19S complex has proteolytic binding sites that attack the target protein. The 20S catalytic unit contains 28 homologous alpha-beta subunits, which are arranged in 4 rings of 7 subunits. Some of the beta subunits have catalytic activity. Amino acids can't be stored in the body, so surplus amino acids that are not needed for protein synthesis are used as metabolic fuel. The alpha amino groups are first removed by transamination reactions, then the nitrogen atoms are converted to ammonium ion or to the alpha amino group of aspartate. The nitrogen atoms of ammonium ion and aspartate are then converted to urea in the urea cycle. The carbon skeletons of the amino acids are degraded to acetyl-CoA, pyruvate, or intermediates of the TCA cycle. Mammals are constantly scavenging amino acids for their carbon content and in the process producing significant amounts of ammonium ion. The ammonium ion is toxic and cannot be tolerated in high levels. The urea cycle converts the toxic ammonium ion into a relatively non-toxic product, urea. The major input into the cycle includes carbon dioxide, ammonium ion, and a nitrogen atom from aspartate, and the only net product is urea. 3 molecules of ATP are involved in the cycle, and 4 high-energy phosphoanhydride bonds are cleaved. Notice that this is a catabolic cycle, yet it requires ATP. The take-home lesson is that the catabolism of compounds that are rich in nitrogen often does not yield a lot of energy. In this slide, the urea cycle in blue on the left is fused to part of the TCA cycle in black on the right. These cycles interact when aspartate is converted to fumarate in the urea cycle. A portion of the TCA cycle is used to convert the fumarate back to aspartate. The first step in the urea cycle is catalyzed by carbamyl phosphate synthetase. The product of the reaction is a very interesting compound called carbamyl phosphate. Carbamyl phosphate has a central carbonyl group bonded to an amide nitrogen atom and to a phosphoryl oxygen atom. Both of these bonds have a relatively high energy of hydrolysis, and thus both bonds require energy to synthesize. For this reason, 2 molecules of ATP are needed for carbamyl phosphate synthesis. One ATP donates a phosphoryl group to the product, and the other is used to drive the reaction in the direction of product formation. The second step in urea biosynthesis is catalyzed by ornithine transcarbamylase. One of the substrates is the amino acid ornithine. Ornithine is not one of the 20 amino acids found in proteins. It's derived from glutamate by a short pathway. You may think of it as lysine with one less carbon atom in the side chain. Ornithine is a sparking compound which is regenerated with each round of the urea cycle, one in, one out, just like oxaloacetate in the TCA cycle. Ornithine transcarbamylase catalyzes the condensation of carbamyl phosphate with ornithine with the removal of an inorganic phosphate residue. The release of the phosphate helps to drive the reaction in the forward direction. The reaction product citrulline has the carbonyl and amide nitrogen groups that will become part of urea. Citrulline is another amino acid that is not found in proteins. The third step in the urea cycle is catalyzed by argininosuccinate synthetase. This reaction condenses citrulline and aspartate to form a very complicated intermediate, argininosuccinate. The alpha amino group of aspartate replaces the carbonyl oxygen of the citrulline side chain. That alpha amino nitrogen will become the second nitrogen of urea when the cycle is completed. The reaction is energized by the hydrolysis of ATP to AMP and inorganic pyrophosphate. On our energy account sheets, this is the equivalent of 2 ATPs being hydrolyzed to ADP and inorganic phosphate. The argininosuccinate is then cleaved by an argininosuccinase to form arginine and fumarate. The arginine intermediate should be familiar to you because it's one of the 20 common amino acids. In addition to its role as an intermediate in the urea cycle, arginine is also a constituent of most proteins. That means the urea cycle plays a dual role in human metabolism by providing one of the non-essential amino acids in addition to synthesizing urea. This is where the urea cycle fuses with the TCA cycle. The second product of the argininosuccinase reaction is fumarate, an intermediate in the TCA cycle. It can be converted back into aspartate by conversion to malate and then oxaloacetate, followed by transamination reaction, which adds on the alpha amino nitrogen to yield aspartate. The final reaction in the urea cycle is catalyzed by arginase. In this hydrolytic reaction, the sparking compound ornithine is regenerated and the urea is released. The nitrogenous bases, purines and pyrimidines, and their nucleotide derivatives can all be synthesized from simple building blocks. and thus none of them are required nutrients for humans. Unlike compounds with a high percentage of carbon, these nitrogen-rich purine and pyrimidine bases are not degraded by humans in energy-producing pathways. The nucleotides are converted to their component bases, and the bases are partially degraded and then excreted. Recall that the pyrimidines contain a single heterocyclic six-membered ring. That ring is synthesized in a stepwise manner from simple precursors. The first reaction in the pathway uses the amide nitrogen of glutamine and bicarbonate along with 2 molecules of ATP to synthesize carbamyl phosphate. The reaction and the enzyme that catalyze it, carbamyl phosphate synthetase 2, remind us of the first step in the urea cycle. The reaction differs from that in the urea cycle in that it's the amide nitrogen of glutamine, not free ammonium ion, which serves as the source of a nitrogen atom. I digress a moment to eulogize carbamyl phosphate. I think that it's one of the swankiest molecules in all of biochemistry, combining an amide bond and a carboxyphosphate bond in one tiny compound. That's quite something. In the second step of purine biosynthesis, the carbamyl group of carbamyl phosphate is transferred into the alpha amino group of aspartate. The product is carbamyl aspartate. The enzyme that catalyzes this reaction, aspartate transcarbamylase, has been investigated intensely because it's a target for regulation of the pathway. It was one of the first enzymes in which feedback regulation was observed. The enzyme exhibits sigmoidal kinetics, and it's subject to both positive and negative regulation. In addition, it has a very interesting structure. The next step in the pathway, catalyzed by dihydroaromatase, involves a cyclization reaction which forges a covalent bond between the amide nitrogen of the carbamyl group and the alpha carboxyl group. In the following step, the cyclic product, dihydroarotate, is oxidized to arotate by a coenzyme Q-dependent dehydrogenase. The reaction introduces a double bond into the heterocyclic ring. The use of coenzyme Q as an oxidant in such a reaction is a bit unusual. The focus of the second half of the pathway is on converting an organic base into a nucleotide. In order to make this a thermodynamically favorable process, the ribose ring is first pyrophosphorylated. Ribose 5-phosphate pyrophosphate kinase uses ATP to convert ribose 5-phosphate into 5-phosphoribosyl alpha pyrophosphate, releasing AMP as the other product. In the next step, the pyrophosphate group on the number 1 carbon of the ribose ring is replaced with an N-glycosidic linkage between arotate and the ribose. In a pattern that we have and will see ad nauseum, the subsequent cleavage of the phosphoanhydride bond of the pyrophosphate energizes the overall reaction. The mononucleotide product of the reaction is aromatidine 5-monophosphate. We finish this phase of pyrimidine biosynthesis with the decarboxylation of aromatidine 5'-monophosphate to give uridine 5'-monophosphate. That compound only needs 2 more phosphates to become a direct trinucleotide precursor of RNA. We will consider the conversion of mononucleotides to trinucleotides later in this lecture. As we've just seen, the biosynthesis of pyrimidines is relatively straightforward. That's not so for purine biosynthesis, which is much more complicated. In fact, it's so intricate that we'll not force you to learn it in this course. You can consider the details of this pathway to be optional, and I have a very realistic idea of what that means. Parenthetically, it's a darn shame that you don't have to learn this pathway. Why is that? One reason is that I had to learn it, so why shouldn't you? The second reason is that my thesis advisor, Bruce Levenberg, helped to elucidate this pathway when he was a student with John Buchanan at MIT. Instead of seeing and learning the reactions he worked so hard to discover, all you have to do is look at a bunch of arrows. That's humbug. Anyway, here's a summary of what you should know about purine biosynthesis. The pathway takes place at the nucleotide monophosphate level. The reaction pathway starts with ribose 5-phosphate and adds elements in a stepwise manner at the number 1 carbon of the ribose ring until the end product, inosine 5' monophosphate, is produced. There are 11 enzyme catalyzed steps between ribose 5-phosphate and inosine 5' monophosphate. Reactants in the pathway include ribose 5-phosphate, glutamine, glycine, N-formyl tetrahydrofolic acid, carbon dioxide, and aspartate. It takes 4 ATPs to energize the pathway. The reactants are color-coded, and the same color code is used in the final product to identify the source of the various atoms. The enzyme responsible for converting ribonucleotides to 2-deoxyribonucleotides is called ribonucleotide reductase. This reaction does not discriminate between the 4 bases, and it can use any of the nucleotide diphosphates as substrates. The conversion occurs at the 2' position of the ribose ring, where a hydroxyl group is replaced with a hydrogen atom. The ultimate source of reducing power for this reaction is NADPH. Electrons from NADPH are passed to FAD, which is reduced to FADH2. From there, the electrons flow to a protein called thioredoxin reductase, in which a disulfide group is reduced to give 2 sulfhydryl groups. The electrons are then passed to a small protein called thioredoxin, in which the same type of disulfide to sulfhydryl exchange occurs. The thioredoxin passes its electrons on to the ribonucleotide reductase enzyme, which in turn reduces the ribonucleotides to 2-deoxyribonucleotides. The pathways for pyrimidine catabolism involve a number of enzymatic steps. The products include beta-amino isobutyric acid, ammonia, and carbon dioxide. I'm not sure whether the beta-amino isobutyric acid product is metabolized any further by humans or is simply excreted. In any case, the body doesn't get much, if any, energy from pyrimidine catabolism. Purine catabolism also involves multiple steps. In humans, the major end product is a heterocyclic aromatic compound called uric acid. Again, there's not much energy derived from this process. The Bella Italia restaurant in Port Angeles serves up excellent Italian food in a relaxed atmosphere. Plus, Bella herself ate here. Some kind of seafood ravioli. What more could you ask for? The chef says that you can take this food to the bank. Say what? Next door to the Bella Italia is the Itty Bitty Buzz. You can get a triple something with lots of caffeine in here. It's just the thing to wake you up after a big pasta dinner. This store specializes in recycled building materials. It has lots of really interesting artifacts. Just outside of town, as you head back US 101 east toward Seattle, you'll see the Blue Flame Barbecue. They smoke their ribs outside in big barrels. You can smell the barbecue as you drive by. Here's a shopping mall, Port Angeles style. If you want a real mall, you have to drive another 65 miles to Silverdale. But who needs a mall anyway? We have 2 casinos in the county. This one only has slot machines. The other bigger casino east of town offers many more ways to lose your money. By the way, do you think there's any double entendre in this sign? Are 2 heads really better than 1? This shop offers some kinds of kinky services, piercings, and other things I never dared to ask. What is this big-headed thing hugging my grandson? Rosie the rabbit? Or what? Here's a view of the Port Angeles waterfront in the spring. The local merchants and garden clubs plant tons of beautiful flowers to brighten up the town. Because Port Angeles offers a deep water harbor, large ships can anchor here. There's a repair facility that services huge oil tankers and other types of ships. For many years there were 2 options for travel to Victoria. The slower Coho ferry, which takes 90 minutes, and this express ferry that made the trip in about 60 minutes. Recently the speedier ferry was removed from service. Now the Coho is the only choice, but it was a lot more fun anyway. The reflections of the Victoria Express make an interesting and colorful abstract picture in the water. Every year there's a rally of tall ships in the Port Angeles harbor. For a substantial fee you can go sailing on one of these ships. Port Angeles has a sister city in northern Japan called Mutsu. Mutsu reflects Port Angeles in that logging and fishing are the major industries. The citizens of Mutsu gave this bell to Port Angeles. There are many interesting older houses in town, including this one, which serves as the home of the public defender. This colorful duplex is located a block or so from the harbor. Note the interesting composition siding on this house. It's like roofing material. They don't make anything like this anymore. When the logging industry was at its peak, Port Angeles had some very affluent citizens. This house dates from that era. We also have some charming but quite run-down buildings. This garage needs a little work. Did I say charming? Here's an old wall with various kinds of moss growing on it. The interplay of the moss with the concrete blocks makes an interesting pattern. The Port Angeles Airport lies just west of town. The open space allows a good view of the mountains surrounding the town.